Welcome to foodfeedinfo.com. This lecture is a part of BS Poultry Science scheme of studies. In this lecture, we will discuss digestion and absorption of carbohydrates with particular reference to poultry birds. Here are few terms which are commonly used related to this digestion and eating of the stuff. The first one is hunger. Hunger is a physiological desire for food following a period of fasting, whereas appetite is a learned or habitual response to the presence of food. So there is a little difference between these two things. Hunger is basically when we feel hungry. It means we have uh, not eaten anything for a long time, like after fasting. If we eat, that is known as hunger. Whereas the appetite is, if even if we are not hungry, we have taken a lot of stuff, but sometimes a certain dishes which we usually like, they attract us, or we eat more than the usual. So that's basically a response to the presence of feed, which is actually a learned or habitual response. So that is known as appetite. So this is same. Sometimes we take appetizer to increase or to take more food items. Then comes the digestion and absorption. Digestion is simply to break down large food particle into smaller particle so that those particles can pass through the mucous membrane and enter into the bloodstream. So that process is known as absorption. Hypothalamic control of appetite. How appetite is controlled by the hypothalamus. Hypo means below, thalamus is the part of the brain. So hypothalamus have two areas. The one is known as the feeding center, or you can say area or the center. The one is known as feeding center. So it is located at lateral hypothalamus. So when feeding center is stimulated, it causes commencement of eating. So the stimulation of feeding center actually causes an individual to start eating, whereas the other center is known as satiety center. So it is located at ventromedial area of the brain. So satiety center, simply by meaning you can understand satiety mean or satiable. To get satiable, it means you get satisfied. So when this center gets stimulated, it will depress the appetite, so it will cause stoppage of the feed intake. So these two centers, they are regulated by different feed intake theories. We will see in the next slide how these theories work and they affect the hypothalamus. Here are feed intake theories. There are three theories. The one is chemostatic, second is thermo, and the last one is gut feel or stomach feel. Chemostatic theory, chemo mean the chemicals, these chemicals are usually the end product of digestion. Like in case of carbohydrate, sugar is the end product, and fatty acids in case of lipid, and amino acid in case of protein are the end product. When a certain level of blood sugar or amino acid or fatty acid is attained, so that will either cause stoppage of the feed intake or it will induce feeding. Like when there is a low blood sugar level, we usually feel hunger and we go for eating. In case of ruminant animals, the volatile fatty acids are then product of fermentation, carbohydrate fermentation, so that cause the stoppage of the feed intake. If in case of human being, the common example is if we eat sugar before eating the meal, we usually eat less. Whereas if we do not take sugar, we directly go for eating the meal, we eat more as compared to individual who eat more sugar before eating the actual meal. So this is basically due to rise in blood sugar level that cause this stoppage of the feed intake. Then comes the thermostatic. Therm mean temperature and static means stop or uh, a decrease in hypothalamic temperature will induce feeding, whereas an increase in hypothalamic temperature will not induce feeding, rather it will cause stoppage of the feed intake. Say high temperature have a negative impact on the feed intake. The common example is during the summer season when environmental temperature is high, so it causes a rise in body temperature. So usually we eat less during the summer month as compared to the winter month. The last one is gut feel or stomach feel. This is basically related to the capacity of the gut or stomach. So when that gut is filled or GIT is distended due to the presence of the food, no more food intake is possible. So here is the definition of digestion. So it is the process of breaking feed down into simple substances that can be easily absorbed by the body, or we can also define it the process whereby proteins, fats, and complex carbohydrates are broken down into units that are small enough size to be absorbed through the gut wall into the animal body. So in simple term, we can say digestion is basically responsible for breaking large food or feed particle into smaller particle so that they can get easily absorbed in the intestine. Then comes the digestive system. So this system is basically responsible for digesting the stuff. So these are the parts of the body which are involved in chewing and digesting the feed. The absorption is the process of taking digested part of the feed or smaller particle into the bloodstream. So it consists of the processes that result in the passage of feed or small molecules from the lumen of the GIT through the mucosal cell lining the surface of the lumen and into the blood or lymph system. So the overall function of various digestive processes is to reduce food to a molecular size or solubility that allows absorption and cellular utilization of individual nutrients released in the process. So question arises, why do we need to know about the digestibility of the feed stuff? Because if we have a feed analysis, we have the chemical composition and we can compare feed stuff. But keep in mind, feed analysis only tell us about nutrient contents. So it is not a true indicator of its worth to the livestock. Then how the feed will react when subjected to the physiological phenomena occurring in the animal. That feed analysis cannot tell us, and it does not tell us anything about the palatability. So it can only be known when we feed that stuff to the animal and then that stuff is being utilized in the animal. So through digestion, it can only be possible. So the actual feed value depends on its digestibility and absorption. A little bit about the modes of adaptation, which are basically you can say related to the feeding of an individual. So I'm not going into detail because here I will just give a little overview of those modes of adaptation by different type of animals. So the first one is the behavioral adaptation. So that is basically the feeding behavior and it based on the type of feed eaten. So there are three types of modes. The one is herbivores, which usually eat the green stuff, the carnivores, which usually eat the meat or flesh, and the omnivores that eat both type of green stuff as well as the meat. The second is anatomical adaptation. So on the basis of digestive system anatomy, we have non-ruminants, which are also known as simple stomach animals. Then we have the ruminants, which are able to digest large amount of green material through microbial fermentation. And we have the hindgut fermenters. They have the same ability as in case of ruminant animal to digest a large quantity of fiber, but here the fermentation of that fiber is in the hindgut of that individual. So these individuals are not able to get maximum benefit of that fermentation because the absorption side is before that hindgut fermentation. The last one is the avian. Avian have a different type of digestive system. We will discuss in the coming slides what are the differences in the avian digestive system. So on the basis of type of digestive systems, we have monogastrics like horse, rabbit, pig, cat, human, and dogs. We have ruminants that have a chambered stomach, four chambered stomach. Then we have poultry or avian species, chicken and turkey, that are also monogastric, but their digestive system is different than that of the monogastric. And in the monogastric, we have hindgut fermenters. They are also simple stomach animals, but they have the ability to digest large amount of fiber in their hindgut through cecal or colon fermentation. So digestion is the process of preparing food or feed for absorption. It involves both physical and chemical processes. So simply you can say digestion have both physical and chemical processes, and the modes of digestion are through either mechanical way, that is known as mechanical digestion, and it is achieved through chewing of the stuff or mastication of the stuff through teeth usually. And in case of avian species, gizzard is responsible for grinding of the stuff. So muscular contraction of the GIT tract is also responsible for some mechanical grinding of the or mechanical digestion of the feed stuff. The second is a chemical action. So digestion can be accomplished through different chemicals, which are usually the enzymes and acids. In the GIT tract, particularly in the stomach, there is a production of hydrochloric acid, and there is also a production of bile salts in the small intestine, and there are the enzymes that all help in chemical digestion of the feeds. The third one is a microbial digestion. So microbial digestion is present in almost in all species, but the extent and the rate of this microbial digestion is different. In case of ruminant animal, the major you can say digestion is through microbial fermentation. This is also enzymic because these microorganisms, they secrete enzymes that digest the that ferment the stuff. So it is brought about by the action of bacteria, protozoa, and fungi. And in case of human being, Zika have these microorganisms, but that fermentation is not too much. And in case of hindgut, as well as in case of avian species, they have a large ceca and colon that is responsible for this microbial digestion. So this chemical and microbial digestion is achieved through enzymes. Either they're produced by the microorganisms or they're produced by the body itself. So enzymes are organic catalysts which are produced by certain cells within the body, which speed up the biochemical reaction at ordinary body temperature without being used in the process. So in the body, these enzymes, they are produced in a form which is known as zymogens or proenzymes. The reason being because if enzymes are produced as such in active state, so they can digest the GIT itself. So to protect that process or to protect the GIT tract, these enzymes are produced in inactive form. So these zymogens are usually the inactive form of these enzymes. They get activated when feed is there and a certain stimuli is there. Usually that is the pH of the GIT that convert this inactive enzyme into the active enzymes, or in some cases the one active enzyme is responsible for converting another zymogen into an. So general part of the digestive system in almost in all species, they are mouth, esophagus, stomach, small intestine, having three parts: duodenum, jejunum, and ileum, and then cecum, the portion at the junction of small and large intestine, then large intestine having rectum and colon, and finally the anus, and in case of bird it is cloaca. So when we define this alimentary canal or digestive system, so simply it is a tube which started from lips and end up at the anus. So tube from lips to the anus, and the accessory organs which are attached with the digestive system, that's liver and pancreas. Non-ruminant digestive system or simple stomach digestive system is divided with respect to the functionality of the cecum and colon. So there is only monogastric or simple stomach, which usually have a very little cecal or hindgut fermentation. And then we have a non-ruminant herbivores. They have a functional cecum. They are also known as hindgut fermenters. And the third one is the poultry, which is the subject which under discussion now. So it's a simple stomach, but the birds, they have a very large ceca and complex large intestine. So ceca are the two blind pouches at the junction of small and large intestine. So if you look at the digestive system of the poultry, so the bird, they have beak instead of you can say the lips and cheek, and then they have the esophagus, then crop, and again the esophagus that empty into proventriculus. Then there is a gizzard, and then duodenal loop or the start of the intestine and first part, then small intestine, and then comes the ceca, and then the large intestine, and finally the cloaca. So briefly, the avian digestive system have some different organs attached to this digestive system. So let's start from the mouth. So in case of chicken or in case of poultry, there is a beak instead of lips and cheeks. So they are replaced by the beak. Beak is basically the prehensile organ. Birds usually pick the stuff through their beak, and there is no teeth, so ultimately there is no chewing in case of poultry birds. The chicken, they are usually the omnivores, and they eat different type of stuff, but in case of poultry bird, the taste is limited. The reason being, the taste buds, they are present on the back half of the tongue. So birds, you can say that they are not able to differentiate the taste. Salivary amylase is present because there is a production of saliva, and salivary amylase is present. Then there is the esophagus that empty into the crop. So crop is simply an expandable storage compartment that is located at the base of the chicken's neck, and feed remain here up to even 12 hours. So it is a diverticulum of the esophagus, and its main function is the feed storage and soaking. So microorganisms which usually come with the feed, they also stay over there, and lactobacilli mainly present and predominate in this part of the digestive system, and they produce the lactic and acetic acid as an end product of their fermentation. So here foodstuff, they get moistened and softened. There is a very little digestion in this part or in this compartment. The food then moves to the proventriculus. So proventriculus is basically the bird stomach where digestive enzyme and acid is produced. So here the mixing and mixing of this feed also occurred. The proventriculus is known as a glandular stomach with a little motility, and then food passes due to the esophageal contraction. So here digestive juices, they are added which contain HCl as well as this proteolytic enzyme pepsin. So from proventriculus, the food passes to the gizzard. Gizzard is a muscular organ with internal ridges. It contains stones or grit, so that helps in grinding of the feed stuff. So feed usually get mixed with the enzymes, and physical grinding occur in this part of the digestive system. Function performed by the gizzard is similar to that of the teeth. And also choline, similar as a keratin type material, so that cause hardening of the surface of the gizzard. So it will protect the surface uh, through this acid attack, because acid produced in the proventriculus that pass on to the gizzard. Uh, Due to the presence of this proteolytic enzyme, the proteolysis occur in the gizzard. Then comes the small intestine. Small intestine is connected with three pancreatic and two bile ducts. They open into the intestine. There is no lactase activity. So in case of bird, the lactase activity is almost nil. So from small intestine, the feed passes to the ceca. Ceca are two blind pouches at the junction of the large intestine. Then from ceca, the material passes to the large intestine, to colon, and then to the cloaca. So usually 2.5 hours is the digestion process in case of laying a broiler birds, and it is up to 10 hours in case of non-laying birds. A little bit about grit. Grit is commonly added to the chicken feed to aid in digestion. Uh, that helps in grinding of the feed. So cloaca vent. This is you can say the opening of the digestive system to the external environment. It's a common chamber where digestive, urinary, and reproductive tract opens. So when fecal material is excreted, the vent fold back that allow the rectal opening of the large intestine to push out, and it closing the reproductive tract opening. Then some specialized organ which we can say that differentiate the bird or avian digestive system from other species. The first one is the crop. Then they have the proventriculus. They have the gizzard. They have the vent, where three type of system: digestive system, reproductive system, and urinary system collectively opens. Then comes the digestion process in poultry. So in poultry bird, the saliva production is there uh, in the mouth, and that saliva contain this ptyalin. Is basically the amylase. It contains amylase enzyme that help in starch digestion. So here, due to the presence of ptyalin, the starch get hydrolyzed and it is converted into glucose. Due to short stay time, the hydrolysis of this starch is incomplete, and due to the absence of teeth, there is no mastication in the mouth of the chicken bird. So saliva is almost 99% water, and the leftover 1% contain mucin, inorganic salt, and two enzymes, the alpha amylase and lysozyme. So animals like horse, cat, and dog, they lack this salivary amylase. Lysozyme basically hydrolyze the beta-1,4 and acetyl glucosaminidic linkage of the repeating disaccharide unit in the polysaccharide of the cell wall of many species of bacteria. So simply you can say, okay, this lysozyme cause killing and dissolving of the bacterial cell due to hydrolysis of this glucosaminidic linkages that are present in the cell wall of the bacteria. So this lysozyme have an antibacterial activity. So salivary amylase helps in this carbohydrate digestion, as it digests the starch, glycogen, and other related poly and oligosaccharides. So if there is only alpha-1,4 glucosidic bond present, so that are broken down by this amylase. So this enzyme hydrolyzes alpha-1,4 glucan links in the polysaccharides, uh, and that result in the production of glucose. And uh, if there is a alpha-1,6 side chain, so we have a structure that is known as amylopectin. So amylopectin are not attacked by this alpha amylase. So simply due to the hydrolysis of the starch through this amylose, there is a production of glucose and maltose. But amylomectin, if it is present, then the product include a mixture of branched and unbranched oligosaccharides. So that is known as limit dextrin. So in which alpha 1,6 bonds are present in abundance. The crop is an extension of this esophagus. I have already mentioned it is normally a reservoir for storage. Food materials stay for different length of time depending upon its particle size, amount consumed by the individual. If particle is small, so that pass on quickly. If the amount is less, then it material passed on quickly. Then quantity of material already present in the gizzard, so that can affect its stay time. So storage, and the second is softening of the feed occurred because that contain the mucus type material that coming through the mouth. So the continuation of amylase tiling activity is there. As such, there is no enzyme production in the crop. Then comes the preventiculus. So it's a glandular stomach. So in preventiculus, there is no carbohydrate digestion enzyme production. So no carbohydrate separating enzyme in the preventiculus and gizzard. The HCl of gastric juice has some ability to hydrolyze some sucrose to glucose and fructose. Then in the large intestine, it goes from ileum to the anus, and it includes the cecum, colon, and rectum. So at the junction of the large and small intestine, there are the ceca that are responsible for microbial fermentation. The major functions of large intestine are to absorb the water and also to absorb some vitamin and minerals. So there is no digestive activity in the large intestine. Normally here the absorption occurs, but in the ceca, due to the presence of microorganisms, there is a fermentation or microbial digestion, you can say, but you actually it is a microbial fermentation. Here's a complete summary of all the digestive activities. Here you can see the region starting from mouth till large intestine, then what is produced in that region and what are the enzymes in that fluid or juices produced in the regions, and what are the substrate, and what are the actions of those enzymes, and what are the end product of digestion. So you can get a complete guide about these digestive activities performed in the small intestine, particularly here with major reference to this carbohydrate digestion. There is the production of salivary amylase in the mouth. Then there is the production of pancreatic juice that contain carbohydrates and in the form of this pancreatic amylase. And then from the intestinal juice, there is a production of disaccharides, sucrase, maltase, lactase. So all they help in the digestion of carbohydrate and converting that polysaccharides or oligosaccharides or disaccharides into the monosaccharide units. And finally, there is the production of glucose or other monosaccharides like fructose and galactose in the small intestine where they get absorbed. If there is a fiber material or the fibrous carbohydrates are there like cellulose, so that get fermented in the ceca and there is the production of volatile fatty acids. But due to the lack or the absence of the absorbed side, there is a little absorption from the ceca, but usually that end product are not available to the animal for utilization.